What's up, guys? Hey, a couple people made it. What's up? Hey, hey guys, how y'all doing?
Good.
Oh my gosh, man, my internet died at the house. That's why I hadn't been able to do anything for a while. So, but I got it hooked up last night in the rain, and my wife is doing her class. She's teaching it for Memphis City Schools. So we're running crazy, but y'all been doing fine. I think y'all fine. A little people had trouble uploading some stuff. I think some questions possibly, but anyway, I just want to go over chapter 24. I thought about showing y'all, kind of making sure y'all understood what we're looking at for the final grades, right? And talk to y'all. But I am recording this session so y'all can go back, and if I go, I'm going to lecture over the urinary system, but y'all can go back over it if you want to. It's there for you to look at. But I really appreciate y'all being able to come in on short notice today. We'll give everybody a second before we get started, but I hope I can get us through it pretty quick. That worked pretty good for that last test, at least having the videos online that y'all can look at, right? Of me going through the chapters. I know several people texted me and said they appreciated it, so I think that's a good idea. Everybody doing okay? All right, nobody's gonna do video, huh? All right. See, if I can see y'all, I can see if y'all having trouble with something. If you're not, if I can't see your eyes, then I'm gonna assume that you understand all completely and you're gonna make 100 on it, right? And Quentin there, man, he ain't moved, man, in like 5 minutes. I'm worried about Mr. Morgan. He ain't moved at all. That's a cool picture, man. I like it. Anyway, I'm just picking. Okay, I'm gonna go into the class real quick and show y'all kind of where we're at. I've got the PowerPoints. This should be better than the scrolling we did before. I actually got easy on some of that. That got me sick. But anyway, I think this will work better. I'm in my office at school, and I have PowerPoint working. My PowerPoint was not working at home. I'm gonna share my screen. I'm gonna show y'all kind of how I'm gonna do the grades. I think I may have showed y'all this already, but let's just look at it real quick. Oh my gosh, I got 1,000 things up. Let's try this one. Okay, let's see. I was actually in lab here. Everybody catch the videos, the lab stuff, right? It went up last night. So I think Angel Nickens has done a tremendously great job helping setting up this online. She had done the work for our lab book and had a lot of these pictures. So if you look on your lab, we have the cat dissection up, and I just had one of the students ask me about, you have to print this out if you got a printer, or write it out and take a picture of this cat dissection video worksheet and then upload it down here at the bottom. Okay, but I may meet with y'all just about lab at some point just to make sure everybody's okay, because that lab is next Wednesday and it counts just like on midterm did, counts a third of your grade. But there's a practice test in there, and y'all can take it over and over and learn the material. Don't cheat. Just use the practice test and let it teach you the stuff we want you to know. Then go in there and take the final on next Wednesday. You have limited time, so you don't have time to Google it up, we hope we've cut down on the cheating, is our attempt. I'm going to go to our class real quick, our lecture. I did send out, and thank y'all for doing it today. It was just a good time for me to do it. I got to get home, watch NFL draft tonight at 6:30. I haven't had any sports for 6 weeks. I'm in detox or some kind of Betty Ford clinic or whatever. But let me show you on my side. I can show you a little bit easier how I'm doing the grades. I'm going to pull y'all out of the way. If you look right here, you can see that these online chapter dynamic study modules, we had 100 of them. They count 10% of your grade. I threw in that discussion board we did the first week. Number 2, week 1, I made it count as a weekly quiz. And I've got it set probably with 1 or 2 drop grades right now. I could look in here. Hold on. I have it set right now with 1 drop grade. At the end of the semester, when we get through with lecture next Friday, next week, I'll do the grades on Monday, Tuesday. I'll probably up that one. That's 10%. The mastering is 10%. I've got 1 drop grade on that, probably have 2 on that. I'm going to have more drop grades on the DSMs because there's so many of them. But we're going to have drop grades there. And then I moved up test 1 to 25%, test 2 to 25%. The test 3 that y'all, quite a bit of Googling was going on, was 10%. The one that we took that was not as good, that I may end up curving later on, I didn't curve it. The high score was 92. But right now I've got it in there. It's 10% of your grade. And then the final next week will be 10% of your grade. So that breaks it down, I think, quite a bit. And we used most 50% was stuff we already had done, which I think is fair. I would warn y'all, I'm just going to tell y'all that I spent probably 5, 6, 10 hours the last 2 weeks talking teachers off the cliff. Lisa Strong called me so upset about cheating in her majors in A&P. And Angel Nickens, boy, she got so mad. She was up to midnight last night. She got mad, made out a whole new final for her. So please just tell your friends to do your work and you'll be fine. But don't try to Google it. We just makes us make it the time shorter and the more short answer. She's actually put a big discussion question on the final, probably one that I will have too, on the female cycle. Describe the female cycle. And we'll show you that. I'll do a Google next Monday, Tuesday, Wednesday, probably on that last chapter or so. Okay, real quick. Now I'm going to go to PowerPoint now. But does anybody have a big question about that one? I think that helps, right? That kind of shows you what I want to do, right? Everybody okay with that? Everybody feel okay? And
Yes, sir.
Okay, thank y'all. Y'all can leave your speaker on. At least y'all can holler at me if you don't want to look at me, or at least let me look at you. I'm sorry, y'all got to look at me. This is actually a cool picture. Take a look. This is one y'all haven't seen before. I think this is me going up Angels Landing in Zion National Park. It's about 3 miles uphill, and right here, this change, it's 800 feet left, 2,000 feet left, 4,000 feet right. So you got to walk across that little narrow part there with the chain and hold on to it for dear life, which I did do. Wished I was out there soon. I'm about to get sick of being sitting in the house. All right. Okay, let me go to PowerPoint. Here we go. Can y'all see? There we go. That's a little bit better, isn't it? A little clearer. And I'm not going to have to scroll. I can flick like this, so we won't get dizzy. Everybody good? Thank you, Haley, for letting me see you. So I can leave. So if you do start crying, I'll know to slow down, right? Or if you throw something at the screen, I'll know. And so I mean that I need y'all to talk to me. Everybody else, if y'all want to leave your speaker on, I think y'all all muted, but y'all welcome to turn it on and y'all can ask questions because I'm going to record this. And I think it's good if y'all ask questions and I can answer it and we can have some real feedback. Okay. I will tell you too that I plan on keeping this recording so I can put it up for next fall. So if somebody goes, I don't understand 24, I'm going to tell, or next spring, I'm going to say, go back. I'm going to show it to them on Facebook. I mean, on YouTube, where they have a link to it like I did with y'all. Did y'all find it okay? The ones that didn't get to see it? Or did y'all watch it over? Did it look okay?
Yes.
Thank y'all. Thank y'all. All right, here we go. Everybody ready? All right, we're going to do it. All right, so I'm going to do this test very similar to the last one. So the final will be very similar to the one that y'all did. The last test y'all had, it was a little bit harder with shorter time. And there'll be some short answer questions. I think I've been doing this so long, I'm pretty sure I'll give y'all most of the short answer questions as we run through this. Okay? All right, here we go. So, and this is actually, I think, a very simple system to me. It makes a lot of sense how it works. But the kidney, basically, let's start with this. We're going to jump a couple of slides to start with it. The urinary system, real simply, is made out of 4 pieces here. You have a kidney, 2 kidneys, one on each side, right here. Y'all can see it. The ureters that carry the urine from the kidney down to the bladder. Then you have the urinary bladder that stores the urine, and then the urethra that shoots it out, out there and goes out to their body. Okay. So if I were to have a short answer question, like list the 4 components of the urinary system, you would say, kidneys, ureter, bladder, urethra. Okay. That's pretty cool. Now, how does this thing, so what is the purpose of this system? Okay. Well, here it is again. The kidneys are going to filter the blood for metabolic waste products. Okay, that's the main thing it does. It filters out mainly this nitrogen waste products and excess water and ions and so forth that we need to get rid of. And in doing that, it maintains the fluid, the electrolyte balance of the body and the acid-base pH of the blood, and it controls blood pressure. So it does a whole bunch of stuff. Filtering out waste products is number 1, controlling the acid base of the blood, and controlling blood pressure. Real simply on that one, y'all remember us talking about this. When we let water out, pressure goes down like letting air out of a car tire. And when we add air to a car tire, the pressure goes up. So our kidneys can control blood pressure on the cardiovascular system by really releasing urine. Pressure goes down, And when we don't urinate, we hold it in, then the blood pressure goes up, the volume of water in our blood goes up. And then by excreting hydrogen ions and buffers and stuff, it plays a big role in the pH of the blood. And then we just have a lot of electrolytes, ions that we have to get rid of anyway, sodium, potassium, and some of those have to be excreted anyway. Okay, so that's kind of the main purpose of it. Its components then is the kidneys, these two ureters, one bladder, and the urethra. Remember in the urethra, the male goes out in the penis, and the female, it is, it is a tube opening above her vagina, right? So we talk about that, so that is below the clitoris of the female, and above the vagina is opening her urethra, okay? The length of the urethras are similar in the male and female, interesting enough. And so the dreaded catheterization by men is no fun for women either, okay? So anyway, same diameter tube there, okay? So the bladder stores the urine, and then we excrete it out the urethra. Pretty straightforward, okay? Once again, it regulates things, and here it is. It relegates metabolic waste by eliminating urine, controls the electrolyte balance. The osmolarity is the measure of the dissolved solutes in the solutes in the water itself, water being still the main component of urine, and that can be eliminated, and some of it can be reabsorbed. And it controls the pH of the blood by secreting hydrogen ions or not secreting hydrogen ions. And if y'all remember, bicarbonate is the buffer that the main blood, way we carried carbon dioxide in the blood, but it also is the main buffer in the blood to control pH. And so we can excrete those and not excrete those. As I have now started the PowerPoint, I just thought about something. Somebody email me later tonight, and tomorrow I'll list, I've got a picture of another book that I actually like that lists all the things in the blood, all the ions and stuff, and all of the ions in urine. I think Ammerman does a good job with her book, but she doesn't have a table that shows that very clearly. Very irritating. I just like that. It just shows all the numbers. I'm kind of a numbers guy, but it's kind of like looking at your grade sheets. You can see all of your grades there. Very similar to that. I'll upload that. I actually usually borrow it from another book for y'all. A couple of random things it does. It can control blood pressure, obviously, by secreting water, but it also produces the hormone erythropoietin, which is the hormone that we mentioned on the endocrine chapter that causes the bone marrow to make red blood cells. And so this is back when I was a young man, back in the 90s, when I started working here 30 years ago, I actually almost bought a stock. I didn't buy the stock in a company called Amogen at the time. In 91, 92, they had about 200 employees. Today, they have like probably 100,000. And their first their first billion-dollar drug was erythropoietin. They bioengineered this hormone, and it's given to all people that are having kidney failure, because the main problem with kidney failure, besides not being able to filter your blood, you become anemic because you don't make the hormone erythropoietin. So wished I'd bought Amogen and held it. I would probably not be teaching anymore if I had kept that stock. I'd bought that stock back in 92, 93. That I didn't, but that's a cool drug, and that's still a biotech company called Amogen that some of y'all might find interesting. It can also detoxify some things, right? It plays a role in that, activating vitamin D, and it can make some glucose occasionally, but that's pretty much the main functions there. We've already saw the locations of them. Remember, the adrenal glands are located above it. It makes some hormones up there. We talked about it. You can see the adrenal gland here that makes the hormones, right, that can control the kidneys. Remember, we talked about that one, adrenocorticotropic hormone and some of those others, right? We'll talk about that. We'll come back to that. Okay, now the kidney itself is surrounded in a capsule, and this is that chainsaw picture. It's not too important. But if we look at it, let me get you a picture of it. I love that we don't have a lot of pictures here. I like pictures. But if you cut it open, and this is what we'd have done in lab if we'd have dissected it, but y'all did it on a virtual lab. If you cut it open, you can see that it has three distinctive layers. It has this outer layer that I'm rubbing right here with the area called the renal cortex. This layer under it that's called the renal medulla. And the medulla actually goes from here to here, and it has in it a structure called a renal pyramid. So if you turn your head a little bit and have a few drinks, you can see it sort of looks like a pyramid if you were drunk. I'm not sure if I see that, but it's there. That's the medulla. And then this yellow layer on this drawing is called the renal pelvis. So on our test, this would be a big question, could be a fill in the blank. The three regions of the kidney are the cortex, medulla, and pelvis. Okay, now how this baby works is out in the cortex, you have about, I think it's 100 million, you have, 1 million structures called nephrons, which are these little tubes over here, the yellow tube here. And this is blowed up. That is that loop right there blowed up, that whole section right there, just that one little block. So in this cortex medulla, is a structure called the nephron. And we actually looked at it because it has that structure called the glomerulus, and we drew that for simple squamous epithelium in A&P 1 last semester. And you can see that this series of the whole set of tubes is called a nephron, and it goes up and down, and it loops down into the medulla area, and then loops back up and dumps the urine into these collecting ducts. So I call this the rivers of urine. These little filters filter the blood, the urine collects in the yellow tubes, the renal tubules, and they dump into these calyxes. This is called a collecting duct, and it dumps into a minor calyx that dumps into a major calyx that then goes out the ureter to the bladder. So this is literally the rivers of urine, okay? And so
And that's the nephrons?
that little part right there is the nephron. I've got a better picture in a minute for you to draw, Haley. But yeah, that's a nephron.
That's what the rivers of urine is the nephron?
That's where it's produced.
Okay.
So in other words, it's just, and this, I'll say this, and this is, I think, a lot easier than say respiration, but the catch here is you've got to catch on that this little set of water hoses is where we make urine. Okay, now let me show you, since you said that, I'm going to jump a little bit, and this may be an error, so bear with me, but let me just show you something. I think here is actually what it looks like a little bit better. Here is showing the blood flow coming in. Okay, and we'll go over this real quick, like renal arteries, segmental arteries, interlobar arteries, arcuate arteries, interlobular arteries, and here is the afferent arteriole going into the glomerulus. This is a leaky capillary bed, and all the water, it's like, think about, and I'm going to tell you when I use this example, and it's kind of gross, it's shock biology, but imagine eating a bunch of pizza and a bunch of salad and overeating and getting sick and running and throwing up, but throw up in some pantyhose. I know that's gross. What would squeeze through the pantyhose? Somebody? Would pepperoni go through? No.
The liquid.
Hey, thank you. There you go, Quinlan. There we go. There we go, Mr. Morgan. Thank you. So the water goes through and all the electrolytes, and it goes in this tube. You with me? And as it goes up and down here, we pull the good stuff out, put it back in the blood, and all the waste we want to urinate out ends up staying in this distal convoluted tubule, going in the collecting duct, going all the way down, dumping into the renal pelvis, and going out the ureter to the bladder. And that's it. Now we got details to look at, but that's the big punchline, is the blood comes in to 1 million of these little filters. Here, the water goes out, and all the stuff dissolved in the water stays in the tube, and all the blood, the red blood cells and white blood cells and large plasma proteins, they come back out this efferent arteriole. Look, come back around here, grab the good stuff, get the good stuff put back in, comes back out, reverse now. You with me? Interlobar veins. And what's the next slide? Here we go. Well, they didn't do it. Never mind. But then it reverses that order out for veins. I know there are a number. I didn't see them. Sorry. 9, 10, 11, 12, 13, renal vein. Now the blood's been filtered. Is that not the coolest thing ever? Really is. Now, let's see if y'all learned something this semester. What if you got high blood pressure like Robin does sometimes? Then that high blood pressure makes the pressure high on this blood coming in, and it blows this little capillary bed, and some big chunky stuff, getting some sausage and pepperoni to get in my blood, right? Or big things fall through, and that's when we start to see renal failure due to high blood pressure. And so people who tear up their glomerulus have to be put on dialysis machines to filter their blood. That's pretty cool, isn't it? So I'm going to back up. I'm going to back up to our first picture. But yes, that's it. Somebody asked it. Somebody said it. Haley, I think, said it. And I think Mr. Morgan nailed it, because that is what's going on here. So these little nephrons, the nephron is just the word for the glomerulus and one complete set of tubes that dump into a collecting duct. And they dump into these minor calyxes here that lead into major calyxes that go out to the bladder. Now, kidney stones is when we get this liquid crystallizes and you form these real sharp crystals, and they tear all this in here, and it's really painful. And that's why they say that kidney stones are more painful than childbirth. So women who have had natural childbirth, 9 out of 10 would rather have a baby before they have a stone pulled out their bladder. That's pretty bad. Okay, so here is the blood flow in, and we can have some multiple choice about this: renal artery, segmental, interlobar, arcuate, interlobular, and then it goes to the afferent, to the glomerulus, then comes back out efferent, and then this paratubular capillary bed surrounds all those tubules, and here's the veins back out. I think the picture is better. So this paratubular capillary bed surrounds it all, collecting the good stuff back out of this tube. The tubes are not named in this picture. We're going to name them in a minute. This is the proximal convoluted tubule, the loop of nephron or loop of Henle. Distal convoluted tubules dump into collecting ducts. You will draw one of these on the file, if any way possible. If I can make it where I can draw one. We may not draw one, but I would normally make you draw one. We may not be able to on the final. I don't know if I can make y'all draw a sketch on that. I don't know. And as much trouble we have uploading stuff, I just think we'll make it part of the whatever, the short answer questions or whatever. We may do that instead if we do that.
So
Right.
drawing the blood flow?
I usually make everybody draw a nephron, but I'll have a multiple choice about the blood flow probably, okay? But I just think drawing a nephron helps me know the parts, but we just may not be able to do that on our test virtually. Okay. There's the glomerulus. This is actually where we saw simple squamous epithelium, remember, in the kidney back in AMP1. That capsule is also called the Bowman's capsule. And the capillary is very leaky, so water and all the small stuff falls through. Okay, and here's show a little bit better, nephron drawn out a little bit straighter. So here's the Bowman's capsule, renal capsule, proximal convoluted tubule, loop of the nephron, used to be called loop of Henle, distal convoluted tubules that drain into collecting ducts. And they start out in the cortex, dip down the medulla, come back up in the cortex, and these all dump into minor calyxes. Okay, she's going to talk a little bit about the glomerulus itself with these holes in it. It modifies a fenestrated capillary, very leaky, with these made out of these podocyte cells with filtration slits. We actually have a electron microscope up in here, and you can see here are what the holes look like that the water comes through. And so that's going to all collect in this Bowman's capsule to go out the proximal convoluted tubule. So very, very good stuff. Very cool, I think, organ. The renal tubules are simple cuboidal in some of the proximal convoluted tubules with microvilli, because we're going to reabsorb water and electrolytes that we don't want to get rid of. We excrete drugs and other things into this filtrate. Okay, moving on down. Here's the simple squamous of the loop of the nephron. And there was simple cuboidal down here. This is where we saw simple cuboidal epithelium in the kidney slide. Okay, then we're coming back up, got more simple cuboidal, and then it dumps into these collecting ducts. Okay, now another interesting side note is this structure right here. This is kind of important, where the distal convoluted tubule comes up and touches the opening of the glomerulus is called the macula densa structure, or juxtaglomerulus apparatus. And the cells in here are called macula densa cells. And so these are the cells that monitor blood pressure coming into the kidney and reduce the hormone renin that you remember goes down the adrenal gland after it's converted into angiotensinogen to angiotensin 2 and causes ACTH, adrenocorticotropic hormone, to be produced down there. I believe that's the one, excuse me, not that. Aldosterone is produced by the adrenal gland, and that comes back to the kidneys and causes water and sodium reabsorption. Y'all remember that hormone made by the adrenal gland? Here's where this is the hormone renin that makes the adrenal gland release aldosterone, okay, which is one of our big ones, remember? We had cortisol, aldosterone, and the sex hormones made by the adrenal, if y'all remember that. So we got to go back to the endocrine chapter, if you want to, in your book, to go and look back over the adrenal hormone, okay? And I would think that was the big one there. Huge, famous one there. Okay, so we'll come back later in this chapter. It's going to talk about hormone control. But this is where that renin is made that makes the adrenal gland produce its hormone. And here's collecting ducts, nothing much here. They're going to drain in. Now they try to do this here. There are two types of nephrons. If y'all caught this little picture, Y'all notice some of the nephrons only go barely in the medulla, and they make up 80% of those. I'm backing it up here. The slides never go with the text. These are called corticopnephrons, but the larger, longer juxtamedullary nephrons that make up 20% make the real long ones that dip down into the medulla. So just a little anatomy here for our human A&P lab class or lecture class. But so you got the short ones that make up mainly 80% of these nephrons, and these 20% that have the longer ones that dip way down the medulla. And that's just a cool little anatomy. It talks about kidney stones here. I've already mentioned those. Calcium oxalate salts. Here's somebody that's locked down. There's a huge kidney stone there. And when they're there, and those are real painful, and we already mentioned that. All right, now, on the physiology of this, the books usually break it down, and we'll just kind of walk you through it quick, and we'll go a little slower. But you got several things going on. 3 big processes going on. At the glomerulus, you have filtration. You have what's called glomerulus filtration. That's where we actually do most of the filter it all out. All the water comes out, and look at all the electrolytes: sodium, potassium, bicarbonate, hydrogens, sugars, amino acids are going to fall out. Everything small is going to fall out. Then immediately after that, we're going to get what's called tubular reabsorption, where we're going to reabsorb the water, the amino acids, glucose, and the electrolytes we want to keep, okay? And a lot of the water. And most of this occurs in proximal convoluted tubule. You never find sugar in your urine. If you do, that's diabetic. If you have proteins in your urine, that's a problem. So we're not in the business of throwing out waste good metabolized energy products, right? So when we do that, and we didn't do a chem strip in lab because we didn't have lab, but we have those, then they're like 20, 50 bucks at the hospital where they check your urine to see what's in it. And if there are proteins coming out, that's not a good thing. If sugar's coming out, you're probably diabetic. And so that is tubular reabsorption, okay? And then in several places in the kidney and the nephron, we get secretions where drugs are put in, toxins are put in the blood, including the breakdown products of alcohol and illegal drugs. This is why we can take your urine and tell if you've been drinking. We can even take your urine and tell if you've been doing drugs and stuff, right? They can check cigarettes out for like a month on some of this. Some of the compounds will still be in your urine. And so they're secreted out. And here's where that occurs. Now this, actually, one reason I like the Amron book, in the old days, I would say filtration occurs here. And by the way, that's the only place where filtration occurs. So we're separating the water from the stuff. But then we would say most reabsorption occurs here, and most books says secretion only occurred here. Well, look at here. It's a little more complicated now. We have filtration here, but mainly reabsorb here. But we do reabsorption even in the collecting ducts. We can reabsorb water here. And secretion occurs here. Excuse me, I hit the button like a fool. But we mainly do drugs over here. So if you ever worked in a bank or handle money, they'll do a blood test, see if you've been doing any crystal meth lately or cocaine, because if you had, you probably need money, and they're going to be looking for you to steal some money from the bank or the teller at casinos for hard drugs and stuff. But isn't that pretty cool? So really cool. To me, look, it wasn't easy the first time I saw it, so I'm not trying to downplay it. But this versus, say, respiration this is a 3 versus 11 on the 1 to 10 scale, right? Dead evil versus this makes sense to me. So think about it. So you got a million little filters in your kidney, in each kidney, 2 of them, and they just do, their job is to filter this stuff out of the water, out of your urine. And the good things get reabsorbed back into the peritubular capillary bed that goes into these veins that come back out by the renal vein. Okay. Now I'm going to take a second and pause. Does anybody have a question so far?
No, sir.
I kind of like, I think this is a pretty fun chapter to me. It makes sense to me. Okay. It's really amazing.
Okay, so you said that it comes in through the blood and it gets filtered, and then that's like how the water and stuff comes out.
Everything little falls out right here. And then
Okay.
and it's called filtrate, Hallie. And so as it goes down this tube, we actively reabsorb by active transport the ions and sugars and proteins, all the good stuff get put back in the blood. Right? Look, the blood comes in, and that efferent arteriole comes over here and forms this capillary bed to collect all the good stuff back up, and then that's going to come right back out the renal vein. So literally we're taking the blood in, pulling all this crud out, everything little out of it, and pulling the good stuff and putting it back in the blood, thus cleaning the blood.
Okay.
You get it? And that's cool. It may take you a little time to look at it, maybe, go eat supper or something, come back and look at it. But it does, it just makes a lot of sense to me. And I will also, next week, I'm going to try to pick some times where I'm just going to be on, I may just, be available where if you text me and say, hey, can I Zoom you real quick? We can Zoom and ask the very specific individual, tutoring if you need it. Okay. But like, try to do that some. So if you email me and you're just really frustrated about something, just text me and I'll text you back a time. You tell me when's a good time and I'll try to work around you because I got nothing else but this going on. My time is pretty free if internet's working. I'll meet with you on here on Zoom and we can talk. I do think this is one that I think you can talk through. Does that make sense better, Hallie?
It does.
It really is. It's just one of them that you got to see it. And it takes me a time. It took me a couple of classes to see that. But it really is cool. And there's some videos in here. In a minute, I'll probably play one that's really good. Okay. So they have videos showing all this. Okay. Now she's going to go a little bit deeper and go through it again. Maybe this will help as we look at it again. But notice that the pore hole in here is 70 to 100 nanometers. And so that keeps blood cells and platelets and large proteins, those plasma proteins, from falling out. So red blood cells, white blood cells shouldn't be in our urine or in our filtrate. Okay? But amino acids and simple sugars do fall through that hole. And they're going to be reabsorbed. And that's kind of cool. Okay? And then there's some other ones that have a little bit bigger collagen fibers to filter out large things, to block large proteins from getting in. They're even smaller there. So these are just like seals. You know what a seal is? Have you ever seen one of these seals where you can take dirt like people panning for gold? Okay, I tell you what, like your strainers for pasta, right? It's got the little grates, the holes in the middle. So you cook your noodles, you throw it in there, and the water falls through, but the noodles stay in what do you call it? A cauldron bowl, whatever. What is it called? I don't cook very often. What do you call that you drain your noodles in? Y'all killing me, right? Y'all know what I'm talking about? A strainer? Okay,
Trying to count.
Yeah, thank you. That's what this is. So these little holes allow the water to go through, but the big noodles get caught in the metal or plastic in your when you're making spaghetti noodles and stuff. The plasma proteins, remember, are albumins, and their diameter is 7.1, so they can't fall through that hole, which is kind of cool. Okay, so here it is. So here's these slits, right? And you can see now, look, little things fall through, but big things can't leave, like white blood cells, red blood cells, platelets, they can't go through here. Okay, very cool. So water and small solutes are small enough to pass through the filtration slits and enter the filtrate. Okay, now let's play a little video.
Each kidney contains over 1 million microscopic structures called nephrons. Nephrons are the functional units of the kidney, each one capable of filtering blood and producing urine. Urine production begins with filtration. This process takes place across the filtration membrane, a porous structure located inside the renal corpuscle of the nephron. Filtration produces a fluid called filtrate, which is similar to blood plasma without the proteins. The filtrate collects inside the capsular space of the renal corpuscle, and from there enters the renal tubule to be further modified until it eventually becomes urine. Filtration happens at the glomerulus, a ball of fenestrated capillaries located inside a hollow chamber called the glomerular capsule. A layer of specialized epithelial cells, known as podocytes, wrap their foot processes around the glomerular capillaries. Between the foot processes are narrow gaps called filtration slits. If we examine a cross-section of a glomerular capillary, we can also see that between the podocytes and the endothelial capillary wall, there is a layer of extracellular collagen meshwork called the basal lamina. Together, the fenestrated capillary wall, the basal lamina, and the podocyte filtration slits form the filtration membrane. These components of the filtration membrane act as a series of finer and finer sieves, which allow only relatively small molecules to pass through. As a result, Filtration is selective based on molecule size. Water and small solutes, such as electrolytes, nutrients such as glucose, or wastes such as urea, leave the blood and collect inside the capsular space to form filtrate.
Okay, what y'all think about that? That help a little bit? Okay, okay, let's see. No, let me back up, make sure I'm right. Okay, so she's got questions for that. I'm gonna let you walk through her videos here. And here are the things that come through. And by the way, here's a test question. Here are the main nitrogen waste products in urine. The main compound is, obviously, other than water, is urea. Duh, that's where urine gets its name. And it's made out of ammonia from protein metabolism. So as you eat proteins, you make ammonia. It has to be deaminated. The ammonia has to be cut off. The amine group, NH2, is cut off. Remember that amine group is converted to ammonia, and then the kidneys take ammonia and carbon dioxide and make urea, which is not as toxic, but it has to be disposed of. Carotenoid or creatinine is a breakdown product of, oh my gosh, a creatine phosphate being broken down. Remember, that's energy storage in muscle. And then uric acid is a breakdown product of nucleic acid, DNA in our urine and our body that we don't need, that we need to get rid of from the foods we eat. So protein metabolism, muscle use, creatine phosphate, energy storage, carotenoid there, and then uric acid from nucleic acid production or over presence of in our food. So we get rid of some DNA. Those are your main products. And that would make a great short answer question. What are the main components of urine? Water, urea, creatinine, and uric acid. Okay. And then at the filtration here, it talks about the amounts here, expected, blah, 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 nothing major there. She gets really excited about this filtration pressures and stuff. I'll show you this in a picture. There's this graph here showing it. But as this water gets blowed out, you have, of course, the pressure of glomerular hydrostatic pressure due to the blood pressure pushing it out. Then you got osmotic pressure wanting to push it back in, water back in. Then you have this one here called capillary hydrostatic pressure, and that's the pressure of the water hitting the side of this tube, slowing down, forcing the water back in. All that said, you get a net outward flow of 10 millimeters of mercury pushing water out into the filtrate. I'll let her explain in the video. I'm not going to ask a lot about this, but it is kind of cool that you have blood pressure, osmotic pressure trying to pull the water back in. Remember how ions want to go toward water, and then the capsule itself changes the pressure by 10. Let's watch her talk about that. I love the little flow of the urine here. Do y'all like that? I like it.
We already learned that filtration moves water and small solutes out of the glomerular capillaries to form filtrate. Now let's focus on the three individual forces that determine whether filtration will take place. The first force driving filtration is a strong pressure called the glomerular hydrostatic pressure, or GHP. GHP is the force with which blood pushes on the walls of glomerular capillaries. Recall that glomerular capillaries are fenestrated and therefore leaky. So as blood flows through them, the GHP will drive water and small solutes out into the capsular space. It works much like a garden hose with small holes. As water flows through the leaky hose, some of it will flow out. The higher the water pressure, the more water will shoot out. The GHP is largely determined by the systemic blood pressure, and at about 50 millimeters of mercury, is stronger in the glomerulus than in any other systemic capillary. Two forces oppose filtration. One is capsular hydrostatic pressure, or CHP, and the other is glomerular colloid osmotic pressure, or GCOP. The CHP is the hydrostatic pressure of the filtrate as it accumulates inside the capsular space and pushes on its walls. This pressure of about 10 millimeters of mercury tries to push water back into the glomerular capillaries and therefore opposes filtration. The second opposing force, GCOP, is due to the osmotic gradient created by large solutes that remain in blood. This pressure pulls fluid back into the capillaries by osmosis. It is a force of about 30 millimeters of mercury and opposes filtration. Think of the three forces as playing tug of war. As long as the GHP is stronger than the combination of CHP and GCOP, there will be enough pressure to drive filtration. We combine the three forces to yield the net filtration pressure, or NFP. The NFP drives filtration. We can measure filtration by using the glomerular filtration rate, or the GFR, defined as the amount of filtrate produced by both kidneys per minute. The GFR indicates whether the kidneys can successfully clear toxic wastes from the blood. There are many homeostatic mechanisms that control the GFR and ensure proper filtration.
Okay, what do you think about that? That's a little bit more detail than I want you to know, but it's still a pretty good video, I think, showing that, okay? You know, when he's showing us how that works. Okay, now watch. She's got another one here that's going to show us these mechanisms. I'm not going to go crazy on mechanism of control, but I do like the hormonal control. There's our renin, angiotensin, aldosterone. That's the hormone made by the adrenal gland. And there's a review of how it's released. Remember, renin goes in the chemical converts angiotensin to angiotensin 1, and then only in the long epithelium do you get the conversion to ACE. Okay, these hormones have a huge effect. There it is, drawn out a little bit better than renin, going to the kidneys. Going from the kidney to the blood to the lungs. Then it goes to the adrenal gland, causes aldosterone, but it has other effects, increasing fluid intake. Look at that, promotes sodium calcium reabsorption, thereby water reabsorption, vasoconstricts blood vessels, cuts down on the efferent arterioles, so more blood can't leave quickly in the adherent, so it blows more blood out. All that's going to increase the rate of filtration. So fascinating mechanism here on control. She's going to walk you through it here on a video.
One of the most easily adjustable factors that regulatory mechanisms can influence to maintain the GFR is the diameter of the afferent and efferent arterioles. The afferent arteriole brings blood to the glomerulus, whereas the efferent arteriole drains blood away. Think of the glomerulus as a sink that you want to keep full in order to maintain filtration. The afferent arteriole is the faucet, and the efferent arteriole is the drain pipe. Factors that reduce the GFR, such as a low systemic blood pressure, which results in a low GHP, cause less filtration. This is because the sink does not contain enough water. How can you fill it up again? By opening the faucet and plugging the drain. This is exactly what happens in the kidneys. The afferent arteriole is dilated to allow more blood into the glomerulus, and the efferent arteriole is constricted to create a backup of blood in the glomerulus. Therefore, we see an increase in the glomerular hydrostatic pressure and the GFR. The opposite is also true. If the blood pressure and GFR are too high, homeostatic regulation can decrease the pressure in the glomerulus by constricting the afferent arteriole and dilating the efferent arteriole, or turning down the faucet while opening up the drain. Next, let's quickly examine two of the major regulatory mechanisms and how they control the GFR. First, local mechanisms initiated by the kidneys respond when the GFR drops too low or rises too high. These local mechanisms are an example of autoregulation and work by adjusting the diameter of the afferent arteriole. A second regulatory mechanism is hormonal regulation of the GFR, which is part of a larger system that controls systemic blood pressure. Low blood pressure and a low GFR result in activation of the renin-angiotensin-aldosterone system. Renin is released by juxtaglomerular cells of the afferent and efferent arterioles, which results in the multi-step activation of the hormone angiotensin II. Angiotensin II increases blood pressure by multiple cardiovascular and renal effects. In the kidneys, it causes constriction of the efferent arterioles. It also causes reabsorption of sodium ions and water, both directly and indirectly, through the release of aldosterone. Water reabsorption results in greater blood volume and therefore a higher pressure and GFR. Interestingly, the hormonal response to a high blood pressure and a high GFR leads to an even further increase in the GFR. High blood pressure causes the release of a hormone called atrial natriuretic peptide, or ANP, which opposes the effects of angiotensin II and lowers the blood pressure. In the kidneys, ANP causes dilation of the afferent arteriole and constriction of the efferent arteriole to raise the GFR. You might ask yourself why we would benefit from a further increase in the GFR. The answer is that a high GFR means increased filtration and therefore more fluid loss. More fluid loss causes a decrease in blood volume and effectively lowers blood pressure. This doesn't come without a price, as chronically high blood pressure can result in kidney damage.
Hey, what's up? Hang on just a second. I'm zooming. What's up? What's up? Yeah, hold on. Let me move on to another video. Let me show y'all one more. I think there's one more video here. Hold on. 1 second. Okay, everything okay? I'll call you back. I'll call you back. All right, bye, bye. And Ms. Nickens, sorry. Some of y'all had her for live, I know. Okay, what do you think about that? Y'all talk to me. Anybody want to speak? What do y'all think?
I feel like all these names are confusing me.
I totally agree. It is very confusing. But the main thing there is that is from deep physiology right there. And y'all gonna be working in a hospital one day, and you're gonna be spending a good bit of your life adjusting people's electrolyte, altering this pathway so that they can not overwork their body. To lower, I mean, people that have heart problems, we give them drugs to increase filtration so their blood pressure will fall. Yeah, pretty wild. But that video was really good. I think summarizing this auto regulation. And this one, we actually covered this one in AMP1, AMP2 earlier in the endocrine system, because this is how aldosterone is produced. Remember, the main hormone. Matter of fact, I would guess why God put the adrenal gland on the kidney was for that hormone, because it goes around the world to come back to the kidney to do this effect. These two places, all this is at the kidney. But I totally agree with you. All the filtration names are very tough. Like I said, I mean, but I'm not going to go nuts on all that, but I'm going to ask a little bit about it, right? Okay. Epinephrine can have an effect, right? Remember, sympathetic nervous system can have an effect. And so obviously the endocrine hormones can have an effect here. And she talks more about that, but clogging the drain. I think that was pretty good, the video about if you squeeze down here, you're going to cause more pressure to come in. But if you squeeze down the one that the fluid leaving, that's going to also increase filtration rate. So cutting that down slows down filtration rate. By closing it here, it forces it to go out this way. So kind of interesting. I know water hose is kind of confusing me too. Look at that, you know, increase more blood flow that way, right? You get that effect. Squeezing down on this one, opening this up, is going to slow that down because the blood's going to go on out that way. So pretty wild. Here is a nice table talking about that regulation from that video. Okay. Then she talks about renal failure. These are some tests where you can test to see if the kidneys are working right. And then dialysis, very simply, they didn't do a good picture on this, but imagine this. I'll try to verbalize this. In dialysis, they just pull the blood out of your arteries and run it through a series of tubes that has little holes in it. And those tubes are running through saline that has no ions in it. So all the urea leaves the blood in the tube and goes into the liquid in the tank. And that liquid is always being drained out. And then that blood, then it's just put back in. Now it's been filtered. So you have to sit on that thing for 3 or 4 hours sometimes to get your blood filtered enough where you can not be hurt, endangering your body. And so here we are, 2020, and we made a machine as small as the human kidney to do the job of the kidney. And you cannot live forever on dialysis machines. I don't think you can live more than 10 or 15 years at the max. So at some point, if you have kidney failure, you will have to have a kidney or you will most likely die. My buddy that was a retired teacher friend of mine, one of my best friends, he did peritoneal dialysis, and then he finally got a kidney 2 years ago this Christmas. So, and he had to retire because of that, because he got to where he was really needing a kidney, so he went ahead and retired, took disability when he had 24, he had 25, almost, he was like 26 years here, so he had enough years to retire, but he was able to take retirement. Some of the drugs we can do are inhibitors of this, have angiotensin receptor blockers, aldosterone antagonists, things that block it. And these are some of our most effective drugs for kidney problems. Okay. Then she's going to go a little bit in the tubular secretion. I'm not going to go crazy on that. It is interesting that there's a couple ways it can be done. The water is going to go through these aquaporins, so reabsorption can be through in between the cells or actually through the cell itself. And that's kind of a little bit more known. And then, of course, facilitated diffusion is anything going high and low with the help of protein carrier. And all those proteins and sugars and proteins have to be pumped through by active transport. And by the way, these are terms we covered in AMP1, right? That recall is getting us, remember, active transport, facilitated diffusion, et cetera, et cetera. Nothing much here. Glucose, diabetes is when too much sugar is in the urine, it doesn't get pulled out. All these electrolytes are going to come in, going to be pumped around, moved around. 65% of the water is reabsorbed back into the bloodstream. This is where I would give you that handout. I'll load it up. It shows you the numbers. I'm not too crazy about all this. I'm not going to go crazy on this, but it's just showing how reabsorption occurs. Here's our equation. It's real important because remember, this is how carbon dioxide is transported as bicarbonate right here. So carbon dioxide, water forms carbonic acid, and we break it down into hydrogen and HCO3. And there's your main buffer in the blood and the main carbon dioxide transport in the blood. There's the enzyme here that catalyzes that reaction. Carbonic anhydrase is mentioned. Okay. 90% of bicarbonate is reabsorbed. So a lot of reabsorption. I'll get you a table that shows all that. Water goes through these special tubes called aquaporins. That's kind of a cool term. So these are tubes, channels that carry water only very quickly back in and out of the cell for osmosis. And she's just going way deeper, and I want to go on that. I will let her do a little video on that, but I'm not going to go nuts on that, but I will answer questions. I may have a couple of points on it.
More than 45 gallons of water and small solutes filter out of the blood in the glomerular capillaries each day. Of course, we don't produce and excrete that amount of urine. This is because 99% of the water and solutes filtered are subsequently returned to the blood by the process of reabsorption. Reabsorption reclaims water and solutes from the filtrate and returns them to the blood. This process can happen by two alternative mechanisms. The paracellular route, in which substances pass between adjacent tubular cells, or the transcellular route, in which substances have to move through the tubular cells. Reabsorption can take place via passive transport, such as diffusion or osmosis, or via active transport, which uses ATP. The proximal tubule of the nephron receives the filtrate as it exits the capsular space. The proximal tubule is a site for rapid and extensive reabsorption. Virtually all the nutrients and vitamins that were filtered out of the glomerular capillaries are immediately reabsorbed into the peritubular capillaries. Furthermore, there is significant reabsorption of electrolytes, such as sodium, potassium, and chloride ions, as well as reabsorption of bicarbonate ions, which is critical for pH homeostasis. Finally, about 65% of the water filtered is quickly reabsorbed in this initial section of the tubule. This type of water reabsorption is called obligatory reabsorption. If we pause the flow of filtrate for a minute and focus on water reabsorption, you will see that in the kidneys, water follows solute and is reabsorbed via osmosis, a type of passive transport. Water reabsorption happens via both transcellular and paracellular routes and uses channels called aquaporins. The filtrate is further modified by the process of secretion. During tubular secretion, substances are moved from the blood into the filtrate to eventually be excreted. Secretion always uses active transport and therefore requires ATP. The proximal tubule is the site where substances such as hydrogen ions, nitrogenous wastes, and certain drugs are secreted.
Okay, what do you think? Everybody cool? So that's secretion there. And she just walked through it again, showing how these hormones work. Okay, let's watch reabsorption and secretion in the distal. Now remember, the main thing to remember is filtration, reabsorption, secretion. And she's just trying to show you that.
Filtration in the glomerulus, extensive reabsorption and secretion in the proximal tubule, and water and ion reabsorption in the nephron loop. The filtrate reaches the last part of the nephron, known as the distal tubule. The properties and functions of the distal tubule are very similar to those of the cortical collecting duct, so we will discuss them together. In contrast to the preceding sections of the tubule, where water reabsorption is obligatory, reabsorption in the distal tubule and collecting duct depends on the needs of the body and is closely controlled by hormones. Indeed, water reabsorption in the distal tubule is called facultative, which means able to adapt to a need. Two important hormones that control reabsorption and secretion in the distal tubule and collecting duct are ADH, or antidiuretic hormone, and aldosterone. As its name suggests, antidiuretic hormone prevents diuresis, or loss of water in the urine. It is released by the posterior pituitary gland and controls facultative reabsorption of water. In the presence of ADH, water channels known as aquaporins are inserted into the membranes of tubular cells to allow for rapid water reabsorption by osmosis, provided a gradient is present to drive osmosis. Aldosterone is a hormone produced by the adrenal cortex, and it promotes sodium ion reabsorption in exchange for potassium ion secretion. Reabsorption of sodium ions also indirectly drives water reabsorption by osmosis. Finally, if necessary for pH balance, aldosterone stimulates the secretion of hydrogen ions. The hormone AMP, which we previously discussed in the context of GFR regulation, inhibits the release of ADH and aldosterone. In conclusion, the distal tubule and cortical collecting duct are responsible for the selective reabsorption of water and sodium, calcium, and bicarbonate ions, as well as the secretion of potassium and hydrogen ions.
All right, what about that? Comments? Okay, all right, nobody got anything on that, okay. But that's it, but that's about it on that. It's pretty cool to me. I mean, I think we're getting real close to knocking this chapter out. So that's how we reabsorb the stuff. So ADH effects right there. Okay. And here's a cool picture showing kind of a summary. So look at that. Reabsorption of all that. And I've got numbers I can show you later in another picture of another book. Water reabsorption and some secretion of hydrogen ions and nitrogen waste, uric acid, and some drugs into the urine and the filtrate. Then down here, just water, and maybe some sodium chlorine. And then up here, we secrete hydrogen ions, potassium, reabsorb water, sodium chlorine, potassium reabsorbed. And over here is mainly water reabsorption with a little bit of hydrogen ion secretion to affect the pH. So the pH can be affected over here. By tinkling them out, we can raise the pH of the blood. Okay, what do you think? That's pretty wild. All right, last little section, I believe, getting close here on the parts here, is regulation of the urine concentration and volume. So 85% of water is reabsorbed, 15% of water that can be adjusted according to need. Okay, and talks about osmolarity. That's the amount of dissolved solutes in the urine. I'm going to go nuts on that. This is the concentration of dissolved solutes as it go down the filtrate. So it starts out here at 300 milliequivalents, and then it gets really concentrated in the loop of the nephron, then it dilutes again. Then over here is when we suck more and more water out of it to determine the dilution of the urine. Now, a thing about this is kind of interesting. If you've ever been hiking in the desert or whatever, or on a mountain trail like in my picture, then you will stop urinating. Your body will quit tinkling because you need to reabsorb all the water because you're sweating it out. And when we were in Death Valley, they actually had pictures above the uriners in the male's bathroom where it said, look at this picture and tell which one does your urine look like. And if it was real orange, it says immediately drink 2 bottles of water, and it had colors to tell you how much water to drink. And if it was clear, then you were not dehydrated. So we actually, you know, and if we overdrink water, we will tinkle out pure water, lots of water. So pretty cool here, just showing how the concentration of dissolved solutes appear and how we produce diluted urine. Now, I've got a question coming up here, and it's another one that's very hard to understand, but I'm going to make the question real easy. We're fixing to see how this phenomenon called the countercurrent mechanism, it is going to set up a salt gradient out here in the interstitial space. That is the area not in the nephron and not in the blood vessel. And what we're going to see is all this out here is going to be super concentrated with salt. And the deeper you go down, the more salt is out here. And that literally is what's sucking the water out of the filtrate. And this is actually showing how that salt gradient builds up in here. And this is why nobody I know eats kidneys. No culture I know of eats kidneys, because they would be really bad. They have no proteins, they have no fat, they're mainly connective tissues and epithelium, and they're nasty because they have lots of salt in them. Okay, and that's called the countercurrent mechanism. And it's called the countercurrent multiplier. I'm gonna let you watch the video and see if you catch it. She has a pretty cool video on it. There it is. Now this is the concentration in the osmolarity of the interstitial space. As you go farther down, look, it's 1,200 milliequivalents here. That is full of salt, and that's what's sucking the water out. It's kind of crazy. So watch this little video showing it, because I like seeing the actual electrolytes move. So check it out. This is pretty cool.
After extensive reabsorption of water, nutrients, and electrolytes in the proximal tubule, the filtrate arrives at the nephron loop. Let's track what happens to the filtrate as it passes through this portion of the renal tubule. The nephron loops of juxtamedullary nephrons descend deep into the medulla and act as a system called the countercurrent multiplier. Countercurrent simply means that the two limbs of the loop are close to each other and the filtrate in them flows in opposite directions. To help understand how the system works, we'll first focus on the individual segments of the loop. The thick ascending limb of the loop is made of cuboidal epithelial cells. These cells are impermeable to water, but they pump sodium and chloride ions into the interstitial fluid via active transport. As a result, the concentration of sodium and chloride ions in the interstitial fluid increases, and their concentration in the filtrate decreases. Notice how this has created a concentration gradient in the medullary interstitial fluid. The interstitial fluid concentration is about 200 milliosmoles higher than the filtrate concentration all along the ascending limb. This concentration gradient is critical to water reabsorption from the medullary collecting ducts, which we will see in the later animation. Next, let's look at the thin descending limb. It consists of simple squamous epithelium that is freely permeable to water, but not solutes. As the filtrate descends through this limb, water is reabsorbed via osmosis because of the higher solute concentration in the surrounding interstitial fluid. The filtrate enters the loop with a concentration of about 300 milliosmoles, then begins to quickly lose water by osmosis to match the high solute concentration of the surrounding interstitial fluid. Thanks to the continuous loss of water, the filtrate becomes more concentrated as it flows down the descending limb. It reaches a maximal concentration of 1,200 milliosmoles at the hairpin turn of the loop. The highly concentrated filtrate that reaches the thick ascending limb then drives more sodium and chloride ion reabsorption. This is a key event because the pumps in the thick ascending limb will work only if the sodium and chloride ion concentration in the filtrate is high enough. In summary, sodium and chloride ion reabsorption in the ascending limb promotes water reabsorption in the descending limb, and in turn, water reabsorption makes it easier for sodium and chloride ions to be reabsorbed. Now you can see why we refer to this system as a multiplier. This efficient countercurrent setup allows the two processes to amplify or multiply each other's effects. To see the complete picture, we need to add the vasorecta capillaries. They run parallel to the nephron loop, but the filtrate and blood flow in opposite directions. The vasorecta act as a special kind of vascular system called a countercurrent exchanger. As the vasorecta descend, They pick up sodium and chloride ions and lose water because the blood concentration is lower than that of the surrounding interstitial fluid. By the time the blood reaches the deepest part of the medulla, it has a concentration of about 1200 milliosmoles. In contrast, the ascending vasa recta passively pick up water and lose sodium and chloride ions because the blood now has a higher concentration than the surrounding interstitial fluid. In conclusion, the countercurrent multiplier and exchange mechanisms of the nephron loops and the vasa recta accomplish two main tasks. First, they efficiently reabsorb between 15 and 25% of the remaining water, and second, they are able to create and maintain a steep concentration gradient in the interstitial fluid of the medulla.
Okay, wow. Now, I'm not going to make you know all them numbers. I just want y'all to recognize that this salt gradient created by the countercurrent mechanism is what allows us to concentrate our urine. Basically, you're going to drink about 1.8 liters a day, about 2 liters a day, and you tinkle 200 milliliters a day. So you drank about 2 liters a day and you tinkle 1.8 liters a day out. So you basically pretty much input equals output, and your total volume there is around 2 liters a day of filtrate and a little bit less than that of urine. Is that not crazy? That's pretty wild. I'm not hearing anybody. All right. You're killing me. All right. I think that's the last video. Of course, I don't know if I'm going to do the other chapters short. I might just get on there and do it later. But that's showing you where all that's occurring. Oh, we do have one more summarize. I've got a nice summary in here. Let's look at the whole thing one more time. Sorry, but I do like the videos.
In this animation, we are going to combine what we know about facultative water reabsorption with what we learned about the medullary concentration gradient in order to understand how the body produces dilute or concentrated urine. Keep in mind that homeostatic mechanisms are not directly responsible for urine production, but rather for maintaining water balance in the body. However, the amount of urine produced depends upon the amount of water reabsorbed. Remember that water can be reabsorbed in the distal tubule and collecting ducts only if ADH is present and if a concentration gradient is there to drive osmosis. The concentration gradient in the medulla is constantly maintained by the countercurrent multiplier of the nephron loop, urea reabsorption in the medullary collecting ducts, and countercurrent exchange by the vasa recta. Initially, filtrate entering the renal medulla has the same concentration as the interstitial fluid, so no gradient is present to drive water reabsorption. As the filtrate moves deeper into the medulla, the increasing medullary concentration creates a gradient that drives water reabsorption by osmosis if ADH is present. More water reabsorption means less water remains in the filtrate, and therefore a small amount of concentrated urine is produced. In the absence of ADH, no water reabsorption takes place in the late distal tubule, cortical collecting duct, or medullary collecting duct, and therefore more water remains in the filtrate. At the same time, reabsorption of sodium and chloride ions continues, so the number of solutes in the filtrate decreases. In this way, large amounts of dilute urine are produced. Newly formed urine drains from the papillary ducts to the minor calyces, then from the minor calyces to the major calyces, and finally to the renal pelvis. The renal pelvis then drains urine into the organs of the urinary tract, and from there urine is expelled from the body.
Now, the cool thing I like about that is ADH was one of our hormones, remember, from the posterior pituitary, and so the brain tells us to make ADH. It goes down, causes water reabsorption in the collecting ducts, and that's one of the keys to maintain blood pressure. That's just cool to me. Pretty cool. Okay, but that, like I said, these videos are really good. They're all on the study area of mastering too. You want to rewatch them. All right, and then she goes over the countercurrent mechanism again. There's a nice summary showing the whole enchilada again. I'm not going to watch that video again. I think we're good. There's the normal contents of the urine. That's what you find in urine. Urinalysis is when we do a chem strip. We can look at the color. It can tell us a lot. The darker, the more concentrated, the lighter, the less concentrated. And if it's translucent, light passes through, it contains not a lot of stuff. But if you're ever checking somebody's urine, like in my grandmama's, a nursing home, if it's cloudy, that's a sign of a bacterial infection or too much protein in the urine. So it should be clear, not cloudy. And y'all gonna be doing that in the hospital a lot, nurses. It does have some things dissolving in it. Specific gravity is a measurement of the solutes in the water. And so we can do a specific gravity test that we don't do in lab, but we actually have one for demo. You can see we can measure that. This just gets back to our water. You ever go somewhere like Florida and you take a bath and you feel like the water's sticking to you? That's hard. It's called real soft water. It doesn't have a lot of stuff in it. And then ours actually has more stuff in it, and ours has less specific gravity around here. So when you take a bath, it feels like it comes off of you. But it has to do with the amount of dissolved solutes. Soft and hard water is how much these ions are in solution in the water, and it can be measured with an instrument, which is kind of cool. We also can do tests where we see if your kidneys are working right. It's called renal clearance. We can give you inulin, which is a non-toxic chemical found in garlic and anchovies, and they inject it in you, and it should be in your urine in a certain number of days. If not, you're experiencing kidney failure. Or creatinine clearance, how fast it goes through your urine. Another one there, not worried about. And then the ureters carry it to the bladder. The bladder has a muscle called the detrusal muscle, which is the one that causes us to urinate. And we have a urethra sphincter that controls that so we don't tinkle on ourselves. Here's the detrusal muscle around here. There's the external sphincter, which is very important. So detrusal muscles squeeze in and we urinate. Our number one scientific name is micturition. So micturition is a scientific name for releasing the urine. Got a weird story on this. My mother is 78. She's been having trouble about 6 years controlling her urine. She has to wear diapers. She has to put a plastic sheet on her bed at night. They've done all kinds of stuff on her, all these procedures. Finally, she calls me and said they're going to Botox me. Well, Botox paralyzes the muscles. And I was like, we don't want to paralyze the sphincter muscle. You'll urinate all the time. They didn't. They came in and injected into her detrusor muscles. And what's happening is she had this irritable bladder syndrome where her muscles were contracting all the time, and it just made her blow it out. By using Botox on her detrusor muscles, she doesn't have to wear diapers anymore or worry about urinating in her sleep, which is a big quality of life issue that's helped her. This was, remember, our transitional epithelium here and then transitional in the ureter. Kidney stones can fall down here and form here. If they get stuck in here, they have to use a rotor-rooter to go in and get them out. That's real painful. They come in the urethra. I'll let you ponder that one. Okay, then the urethra carries it out to the open area. Boys and girls both have it. In the male, it goes through the prostate gland. So prostate problems can lead to urination problems. So the prostate covers the male here, squeezes down and prevents them from having a nice stream. So if you are dripping, you may have prostate problems. Girls, here is, and I always do the openings on boys, anus, urethra. Girls, anus, vagina, urethra. And it's between the vagina and the clitoris right there. So a little bit interesting anatomy. I had a woman one time stand up and argue with me that she had 2 openings, that she tinkled out of the hole that her boyfriend visited. And I did have to argue with her about that. And I think finally she kept on, I couldn't have class. And I finally asked somebody to give me I bought somebody's makeup mirror and told her to go to the bathroom and check herself out. She later reported to the class that she did have 3 holes. She did not know that one was there. So, hey, great class. We learned a little personal anatomy that day, and it was very entertaining to add to the class. Yes, that really did happen one day. But there is a birth defect where some people only have 2 openings. The girl's urethra goes into the vagina, so that could be a real condition. And I'm glad she found out that she did not have that. Several animals do that too, so they only have 1 opening there. Micturition then, obviously control the sympathetic parasympathetic nervous system can affect that. The reflex, the parasympathetic nerves tell the bladder when it's full, tell the brain it's time to open, time to urinate, and you get the urge to go. The pons and the brain goes and does that, blah, blah, blah. Nice summary here of the whole thing again. I think it's a pretty cool chapter. What do you think? We did that in about an hour and 10 minutes. Questions, anybody?
No, sir.
I did. Sorry about that. I did have a lady just text me. I had some girl asking me about her labs and lab, and she didn't. I thought the instructions were clear, and I was looking at 2, and she was in AMP1, so I had to go back and fix that. Miss Nickinson made a lab for them to do and give page numbers, and y'all's lab that we run off for y'all may not have had page numbers, but hers should have had it. I don't know. That's interesting. I got to ask her. That was AMP1. I think they bought the new lab books this time. That's weird. I got to check on that. Anyway, sorry about that. How, are y'all going to, do y'all want to do the other one about 15, 20 minutes and be done with these 2? Y'all okay with that?
I'm cool with that.
Let me knock this. That would be really nice if we can knock that out, because I may have to do 2 on genetics and reproduction. Okay, so hang on. I'm going to cut this off and get another PowerPoint up real quick. But I do think that's a pretty fun chapter. This other one is not, maybe 20 minutes at the most, and I may even be quicker than that. I may do it in 10 minutes, because I'm probably, y'all probably had enough of me. Hold on, I lost it. All right, why is it doing that? Oh, I pulled the wrong one up. I wanted the PowerPoint, sorry. There we go. Can y'all see that okay? Are y'all still there?
Yes, sir.
Okay. I don't see y'all now. Okay, I don't know how I did that. Let me see if I can find y'all.
I'm here.
I know, I believe y'all. I just can't see even y'all's pictures now. Hold on. Oh, come on, computer. I love technology. All right, never mind. We're just gonna do it. I'll walk through without y'all. Y'all can see me. I can't see y'all. It's fine. This is a chapter I don't do much on. It's just on electrolytes and fluid content. I kind of cut to the chase and give y'all, if you watch the whole video, maybe you'll get the test questions here real quick. All right, the main thing here, I think, is point here is that if you have electrolytes in water, they conduct electricity. So ions will transfer electricity. Sugar and protein, sugars won't. So sugar water won't. Now don't go home and try that and kill yourself, but in general, the more the ions, the better it conducts electricity. So ions are just elements dissolved in water with a charge, okay? Acids release hydrogens, right? Bases are things that suck up hydrogen ions. So hydrochloric acid is going to release hydrogen ions. Carbonic acid is going to release H, and it becomes H and bicarbonate. Bicarbonate was the main buffer in our blood to resist changes in pH. Okay, the pH scale, y'all know, goes from 0 to 14. 7 is neutral, blah, 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 nothing much there. Now here's a good one. You normally have about 70 kilograms, 154 pounds of water. Okay, now look at this. This is kind of cool. So you have about 42 liters of water in your body. Here's where it's at. 16 liters is extracellular fluid. Okay, and 26 liters, 60% is in the cytoplasm. So 60% cytoplasm, 40% extracellular. Of that, 32% of that is interstitial fluid between the cells, and the rest of it is like plasma in the blood, which is kind of cool. So most of your water is in cytoplasm, in between cells, and 8% in the blood. 60-40, maybe a multiple choice question there. Okay. Now, where are the ions? Blue is extracellular, sodium, chlorine, and bicarbonate are the extracellular. Remember sodium potassium pump? Sodium on the outside, potassium on the inside. So the main intracellular electrolyte is potassium, phosphate, HPO4s, magnesium, but mainly sodium being on the outside, potassium on the inside. That's kind of cool. And proteins are big on the inside or in the cytoplasm, intracellular. So kind of cool where the electrolytes are. Y'all know that sodium is so big in our diet, and we eat too much salt, and that's why we have too much out there in that extracellular, and that can cause problems. Okay, nothing much there, just the way we move things. Nothing much here. I'm kind of blowing through it. I'm not a big fan of this chapter. Dehydration occurs when you have not enough water in the plasma, and so we suck the water out of our cytoplasm, and that causes our blood to swell up, and that can cause problems. So check it out. So girl here sweating too much, the water.
Mr. Robinson,
Yes, ma'am.
are we able to see the PowerPoint? Because like right now, I can't see it.
Okay, I need to know that. Yeah, that would have been nice. I thought we were showing it. I thought y'all could see it.
It's just been like your face in the picture for the past couple minutes.
Okay, y'all gotta tell me because this is a really short one. Sorry. Yeah, sorry. You gotta tell me.
You're good.
I couldn't find y'all. I had to go. I couldn't find y'all either. I couldn't see y'all.
Okay, I can see it now.
All right, sorry about that.
You're good. Thank you.
Let me back up. This is really not a big chapter, so it ain't that big of a deal. Come on, computer. Hang on. Okay, now this is, sorry. Most of this is not that big of a deal. This is kind of stupid here. Sorry about that. But it just showed you about electricity and water and the terms pH and stuff. But there's the one I was talking about. Look, there's where your water's at any time. Sorry about that. 60-40, right? Cytoplasm versus extracellular, and extracellular is interstitial space and plasma. Sorry about that. And here is where the electrolytes are. So extracellular are mainly sodium, chlorine, bicarbonate, and then intracellular ions are potassium, phosphate, HPO4, proteins, sulfates, magnesium. And that kind of cool. These are where your electrolytes are. Like I said, they ain't gonna be 5, 10 points on the test out of this chapter. And that's just talking about how you move things. We already talked about osmosis and stuff. And when y'all hollered at me, here is where I was at. So really not much in this chapter. Dehydration, look, this girl is sweating too much, right? And look what's happening. Her blood the plasma is sucking the water out of her cells, and that's gonna cause her brain to shrink, and she's gonna go and pass out. So to fix that, she's gonna drink water, and that's gonna reset this back so she doesn't suck it out of her cytoplasm. If she drinks too much, she will have water intoxication, and this becomes dilute, and it forces it into the cytoplasm, causing her brain to swell this time. So water intoxication does happen. It happens a lot of times when people drink too much water. If they've been running, there've been people run a marathon and drink 4 or 5 bottles of water and go in a coma or pass out. But more common in the South with all of our heat is dehydration due to sweating and burning out too much water out. Cholera, the main cause of death, one of the top 20 causes of death in the world, is a virus that causes diarrhea in little children in third world countries, and they number 2 themselves to death. They die of dehydration by too much diarrhea. We all get our vaccines for that cholera, and we don't see that very often in America. So it's pretty cool. Good one there. I'm glad y'all. Thanks for stopping me on that. And then we actually do make a little bit of water and doing cellular respiration. It talks about that. And this is cool. Where does the water come from? Where do we lose water? Excuse me. Well, and with both of them are on here. We might get water from. We lose water mainly in urine, about 15 milliliters of water a day in urine. Feces has a little bit of water. We sweat out about 600 milliliters, and we lose it in our breath. That's that coronavirus in that we breathe out, water droplets. We drink about 1,500 milliliters a day, so water intake usually equals urine output. Pasta and other foods have lots of water in it, and then we make 250 milliliters by what's called water of metabolism, the water made in cellular respiration from the last chapter that y'all all hated, right? That's my cross thing there. Thank you for praying over that last test. There is an animal called the kangaroo rabbit found in Arizona in the desert in Utah that can live off the water metabolism. So they're little small critters, and they can live off the water they make by cellular respiration. It rains once or twice a year. They will drink on those days, but they can live off of their own water. We can't do that. We have to have water. Okay, and that's talking about the mechanisms, how we control it. And that's showing the drinking when we're thirsty and how all the effects it has on water loss, water gain. That's pretty cool. There are tables to tell how much water you need to drink. Pretty cool. How many of y'all, come on, man, football players, sports players, take IVs and have time to replace some of these electrolytes and the fluid loss. And that's pretty cool. ADH comes back and plays a big role, remember, and and making us release produce urine by opening up the alveolus. A little bit of review there. Talks about dehydration, water. It says overhydration, water intoxication, and water toxicity. I think our lab book mentions the Wii. Back when y'all were, before y'all born, when the Wii computer game came out, this guys out in California did a thing where they called it Wii for the Wii, and they made about 10 lucky contestants come in. They had cases of water. They drank a bottle of water every 5 minutes. The last one to urinate won the Wii. They get down, I think, two people, and a woman goes into a seizure and dies of water intoxication. Everybody loses their job, and there's a dude in California owns a radio station now, probably, that he inherited. He won in the lawsuit over these idiots making people drink water as a contest. True story. It's mentioned in our lab book at the clinical applications at the end of the chapter. Pretty wild. Edema, etc. Now electrolytes talks about this, what we do with them, sodium for action potentials, blah blah blah. Nothing much here. That's sodium. Here's some terms maybe pop up. Elevated sodium in your blood is called hypernatremia. Low sodium is hyponatremia, and that can be due to overhydration, and that's from not enough water. And that can hurt. That can kill you. Potassium's a big one too. We got special names. Hyperkalemia is potassium greater than 4 millivie equivalents, and then below that 3.9 is hypokalemia. One of my worst stories I've ever heard was one of my students were taking AMP2, and at that time they did clinicals at the same time. This was this time of year. She was about to come. She would be finishing nursing 1, and they were I think they were one of the hospitals in South Haven or in DeSoto County. And you, as an instructor, you can never leave the student with the patient at the hospital doing clinicals. Her teacher was with her. They were in a room with a crash cart across the hall. A student from another college in Memphis was doing their 1-year nursing school, and the instructor said, "Give Mr. blah 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 his potassium treatments." And it's a syringe, and the student went in and shot the whole syringe into the dude's central line, thus killing him. This is how we put people to death on death row with potassium. They're supposed to do 5 milliliters every hour, and the instructor was down the hall visiting with the people at the desk. They brought my friend's crash cart over. They revived the guy, give him a transfusion of blood. He woke back up. They never told him that they killed him. He had no family there at the hospital. That girl was thrown out of nursing school. That instructor was fired, and that school no longer can work at that hospital. Don't play around in the hospital with potassium injections, okay? True story. So be aware to remember some of this, that you don't take potassium all at once. Even if a doctor told you to give somebody a whole syringe, that would be murder, okay? Really true story. Not real big fan of those schools that do 1-year nursing, if you can tell. Here's some drugs that can have effect on that. I'm going to skip all that. I'll put any of that on test. Calcium, we know we need that for bones and all, but it also has to be controlled. Not a big deal there. Too much calcium, hypercalcemia, not enough, low, and that can cause problems. Here's a girl with that one with low calcium, and she's having muscle spasms. Chlorine to make hydrochloric acid, magnesium. I did have a short answer question. Last part here, we're about done on acid base and how we control pH. And here's the answer. The main ways we control pH is 3 mechanisms. The main way is the buffer systems, and there are 3 buffers, okay? And then there are 2 physiological mechanisms. So buffer systems, and then these 2 physiological mechanisms, okay? The buffer systems are, let me get them to show you here. First, they're going to tell us where we make a bunch of acids. We make all these acids during cellular respiration. Ketones are acids. Here are the buffers. We have the bicarbonate buffer system right here. They're the main buffer in your blood to prevent shifts in pH. Got to have it. Phosphates also are buffer systems. HPO3, HPO4s here, they can give off hydrogens, and they can go from dihydrogen phosphate to hydrogen phosphate, and thereby grab them or release them, their buffer. And then proteins have this carboxylic group that can release or grab a hydrogen. So proteins serve as buffers. So 3 buffer systems: bicarbonate, phosphates, and proteins. They control shifts in pH due to acids being made by cellular metabolism. When these fail, we have the 2 physiological buffers. Respiratory system, we can actually breathe out more carbon dioxide, and that causes direct control of carbon dioxide. Then look at that, will help raise the pH. We can go back to our equation. Look at this. So if we increase production of carbon dioxide, blow it out, that pushes the equation to the left, grabs hydrogen ions. We're getting rid of hydrogen, so therefore we're raising the pH, because high hydrogen is low pH. So by getting rid of hydrogens, we raise our pH. And so increased breathing raises pH. That's that one. And this next one's real easy. We just covered it. In last case, we can just tinkle out hydrogen ions. We can put them directly in the urine, thereby raising the pH. That'll be a short answer question on our test to see who stayed and listened to the full hour and 25 minutes for these 2 chapters. So we can tinkle hydrogen ions directly in the urine. Okay, acidosis is if the blood gets below 735. Actually, there's a table showing these. Let me go these, the last note, I believe. I'm not going to let you watch it. Y'all can watch that. I'm going to go to the table summary. So I do put a couple of these probably on multiple choice. Look at this. Respiratory acidosis. So this is decreased pH, increased carbon dioxide levels in your blood due to a blockage of air passageways. Haley and Hayden hold their pillow over their boyfriends at night to choke him to death. Maybe he's been vaping and he has decreased gas exchange in his alveoli. And maybe he has brainstem damage and he's not breathing properly. But vaping and strangling somebody leads to respiratory acidosis. To compensate, we hyperventilate, et cetera, et cetera. Respiratory alkalosis is the opposite. And this can be caused by hyperventilation, brain tumor to the respiratory center, or hyperventilation due to high altitude mountain climbing. And that's a real big deal. Metabolic acidosis is decreased pH, decreased bicarbonate, prolonged diarrhea. And this is why we want children with diarrhea to be given lots of electrolytes and Gatorade. Never give babies water, ever. No one wants to drink water till they're a year old. Pure water, give them milk, give them Kool-Aid, give them Diet, give them Pepsi, give them Coke. But don't ever give them pure water with babies. Metabolic acidosis, renal failure, ingestion of poisons, acids, toxins. Okay, and method to compensate is hyperventilate. And metabolic alkalosis is prolonged vomiting. So you get rid of too much pH from your digestive tract, oral overdosing on taking too many large doses of antacids, or excess aldosterone due to a tumor to the adrenal gland. So I just want you to know that what these are, that is lung-induced acidosis or alkalosis, and metabolic means something in the body chemically causes decreased pH or too high pH of your blood. I believe that's the last thing on the chapter other than dehydration, nothing else. That just shows you all the mechanisms there, going back over what dehydration can do to your body. I think that was the last slide. Sorry about the slide. I do have a couple of these terms on our test. I would think that the big 2 is going to be reproductive system that we'll go over next week. And I'll try to do it early in the week because y'all's lab test is on Wednesday. I may wait and do some of it on Thursday. Okay, here's another question. I would like to go ahead and get this test over Friday, but if something happens and I can't get it together, we can move it to, I did get permission for some of the teachers to move their tests to next week. So they're in the week of finals on Monday, Tuesday, Wednesday, and Thursday, but I'd rather just get this over Friday if y'all fill up for it. We will meet next week and talk more about that. Okay, any questions, guys? I appreciate that. We got 1, 2, 3, 4, 5, 6 people. I'm going to put it online for everybody else to watch it. Thank y'all for coming today. Anybody got a question about anything to do with lecture or lab? I know some of y'all are in my lab. Some of y'all are probably in somebody else's lab, but I'll be glad to answer questions if y'all got a question about anything.
No questions over here.
I'm all good.
Okay, thank you, sir, Ms. Murray.
I
Everybody okay?
have a question about lab,
Okay.
if that's okay.
No, please do.
Okay, so I'm just kind of frustrated with lab altogether, with it being online and everything. But anyway, so how exactly are we supposed to be studying for this final? I don't know if y'all are all giving tests the
We're all doing
same
the
or
same
different
thing.
or what, but
We're all doing the same thing, and everybody's
I
taking it
have
on
no
Wednesday.
idea
I just loaded, it
how
got
do I
loaded
study
last
for
night,
this.
and I put a note out, hey, like, well, I don't know if you've seen it, but there is a review, there is a practice lab final. Okay,
Okay.
take it. I would not Google the answers. I would just take it and do my best and check my answers when I get through. And they're pulling from the same freaking test bank as the final. So if you go in there and you just take it till you make it a 90, you're going to make a freaking 90 when you take the other test, or real close. We actually made it really easy for y'all, because I know we didn't get to do lab like I wanted to. And I'm not going to burn y'all in lecture. I'm going to go back in there and do drop grades. I told my majors class this last night, but I've Zoomed with y'all more than I had the others. I'm not going to ruin your life, but I'm going to have a distribution. I'm not going to have somebody go from a 40 to an A, but I'm also not going to have somebody go from an A to a D or C. Okay, I'm just gonna, I'm laying heavy on those first two tests, because I know we took them in person. I know we didn't cheat. But so what that is, I'm telling you, Haley, is I know that lab test is, if you'll take that practice test, it comes from the lab final is only on what we did since Corona. Because see, normally the midterm, like last semester was first half, and the final is second half. So everything y'all did online these last 5 weeks that's all that's on the final. And so she's got practice tests like y'all did. Remember the quizzes y'all been doing? It's going to be like that, but she's pulling from this test bank. And if you take it long enough, now if you want to, I would not try to take it and take pictures and try to make a list of them. Just take it and let it teach all the material. You don't have to go out here and Google the answers. You can waste 5 hours trying to make a cheat mechanism by just trying to print them all out, but let the darn thing teach you the material. In other words, it'll give you, and after you do it about 10, 15 tests, you've probably seen every question on the final. You follow me?
And I got it.
That makes you feel a little better, don't it?
It does.
I should have sent an email out. We made that decision last week. Everybody's doing it. We're all doing it Wednesday, so y'all won't like take it, and like maybe, we thought all the labs are going down next week. All my natural science labs. Chemistry's one day, A&P's one day. So everybody's got to take it the same day, because I know some of y'all still working some, and we weren't going to make you do it at 1 o'clock on Wednesday or Friday. You know what I'm saying? Like a certain time. So you got most of the day to do it. Okay.
Okay.
But I appreciate you asking me that, because I really do. And like I said, Angel, Miss Nickens did a lot of that, but I think it goes right along with the work that we did for our lab book. You know what I'm saying? It should be pretty easy. Might spell a few things, but still, I would just literally go back and just do that practice test over and over again. And she had like a whole page, I'm trying to get my hands up there, a paragraph explaining the test. Matter of fact, let me show you that real quick. You may not have seen that. Okay. That's a great question. I'm glad you don't ever be afraid to ask me a question. I really think y'all done really good asking questions today. Look at this. Hang on. I got to go. Let me find it. Hang on just a second. Here we go. Let me go to lab. AP2. And I just moved it around today, Haley. So you're in my lab, and I hate I didn't get to finish the cat with y'all. That really breaks my heart. But look, here's this week's lab. Where does it on here? Okay, let me find it. I gone too far. Week 4, week 5. Here we go. This is the one we're doing this week on reproductive system, right? Yep. And genetics. Okay. Now watch. Here we go. Here we go. I thought I pulled it up. I know I did. Here we go. I swear I done moved this up.
It's not showing that screen in Canvas.
Okay, let me show it to you. Hang on.
Yeah.
Hang on. Just 1 second. Just a second. I think I went the wrong one. All right, hang on. One second. Still trying to figure all this out. All right, try this one. Y'all see that?
That works.
All right, sorry. All right, make sure I'm in A&P. I got one section of A&P1. Lord, I wished I didn't, because my mind, I get confused. All right, here we go. So y'all are doing the cat dissection today, this week, right? Get my story straight. Yeah, y'all did reproduction already. Cat dissection is this week. Y'all got to print this sheet off and upload it. But look, here is the lab final review right here. Oh, that's the date. That's that right here. Here are the instructions. Right there. So take the practice test over and over again. You will have 45 minutes to do 80 questions on the test. So you need to take it till you know it. You'll have Wednesday to do it. You got to score 80 on the practice to take the other test. That way, at least I know y'all did some work. So even if you're Googling the practice test and trying to use your books and stuff, you still got to make an 80 on that. And then you ain't gonna have time to use your books hardly on the other. Because she's gonna move them around a little bit.
So we can take the practice test multiple times. It's
Yes,
not like a one and done.
Yes.
Okay.
This is kind of the way we do our non-majors biology classes. And it usually, everybody does real good on the final. So I would expect y'all to do real well. Okay. So it's 80 questions there on the final review. You got to make 80 out of 100 to take the this. And on the final, let me see what it is. You're set up with, I'll make sure I got it right here. I'll just look, take a look. Hang on. You have 45 minutes to do 80 questions. Okay. Does that make sense?
It does.
So I think it's very fair. And matter of fact, Angel and I know y'all know Angel and I been friends a long time. We really are making sure that this is not like psychotically crazy. Because it would, and our final in AP2 is really cat anatomy, which is real easy. So if the grades are bad, we'll cover them up. But we don't want everybody to make 100 because we can't get a distribution on that. You know, if everybody makes 100, then it's a joke. You know what I'm saying?
Mm-hmm.
So that's kind of where we're at. Did I help a little bit, though? I think that was pretty good help there, too, wasn't it? You better feel better, Haley?
That helped. It did.
And I should have told y'all that. I should have told y'all like last Friday, that's what we're doing. But I just got, my world has been crazy, man. We've been running wild. And we still, and I apologize. I don't know if y'all figured out, they screwed up summer school. We should have announced registration. So if you've taken summer school, nothing's going to be what you probably want to do. We're not going to do any lectures in person. We're going to do one A&P, one in June on Zoom like this. And y'all they got to have the cameras on. They got to let me watch them during lecture. And, but we're just got a mess for summer because we can't have more than 10 in a room. We are going to do labs in person this summer. We're not doing these labs online. This has not been fair to y'all. We didn't enjoy it. I know y'all, like you said, Haley, I've been I would quote your feelings that you told me a while ago is my feelings exactly too. Very frustrated with the labs online. You know what I'm saying? And it just didn't feel like y'all signed up for that, but we had to do what we had to do to give y'all's credit this semester. Okay. But I'm not going to have anybody, go to an F from a B, because they just didn't like this part. You might go down letter grade, but I'm going to try my best to not have anybody go 2 letter grades who did their work. But I think this final is going to be a lot easier than some of these quizzes probably. Okay.
That gives me a lot of hope.
Okay, well, good. I mean, it is, but I'm gonna not count it as much as I did the other. Hey, I may alter the final grade if the grade's real bad. You know what I'm saying? So I still may. I'm gonna shuffle the grades to be fair. I know what I want to grade, and I expect a 50% of this class should make an A in lab, or 40% anyway. You know what I'm saying? So I'm probably close to that right now.
All right, sounds good.
Any other questions, guys? I really appreciate y'all logging in. I will be available for individual questions if y'all have them. Y'all can, and I appreciate y'all got if you'll go to, I have a Google number. Just don't call like after 6 or after, you know, from 6 to midnight or whatever. Don't call me in the middle of the night or something, but you can use that Google number and it comes straight to my phone. So if you're having a panic attack and you're on the cliff right here and you're about to jump off like Mr. Robin there, give me a call and I'll walk you back down we'll hold the rail together and come back down. So we got 2 weeks. Let's finish this out. And like I said, our lecture test is supposed to be next Friday. And we'll just see. If I have it ready, we'll do it then. We may move it if I can't get the test together. I've got to work on the test. I haven't got it made yet. But if we do, we will meet Thursday night and have a review for the test, lecture test. Is that cool?
That would be good.
Well, we'll do the reproduction like on Monday or Tuesday night or afternoon or something, and I'll have them online. I'll finish the material as soon as I can next week for y'all to have time to look over the lectures. Okay.
Sounds
Okay.
good.
All right, guys, I'm going to call it a night for y'all. That's been an hour and a half. Y'all guys have been nice. Thanks for paying attention, and thanks for telling me I didn't have the picture up. Lord, I'm still learning this stuff. Y'all have a good one. I was working at home, but now I'm at school today. So thank y'all. Everybody safe? Anybody been sick? Any family?
I'm sick of online school.
Okay.
Same.
Same, it's stressful.
Yeah, we got a lot of students that should test positive. We've had some faculty, and they had an Ole Miss professor that died, an English teacher. So my doctor, that I talk about in class, says it hadn't hit here yet good. So we're probably behind New York and these others. Lord, I went to town today and everybody was out. There's 100 people sitting there visiting, so there ain't nobody doing social distancing hardly, and I think that's a mistake. So be careful, okay? All right, I'll see y'all later. I'm gonna call it a day, and I'm gonna get this thing copied so I can get it online tomorrow for everybody. Okay? See y'all later.
Thank you.
Thank y'all.
Thank you.
We're gonna get through this.